As we break down the different steps of glycolysis and the Krebs cycle, you'll see that each reaction within these metabolic pathways involves a different enzyme. In my class, I'm not going to ask you to know all the enzymes. I'm only going to ask you to know some of the really important enzymes, or ones that we refer to as a rate-limiting enzyme. What happens with a rate-limiting enzyme is they're really controlling the speed of the reaction, because what tends to happen at a rate-limiting enzyme is some sort of bottleneck. And based on negative feedback, they'll control the speed of the reaction. So very often in metabolism, if you have a lot of ATP, or high energy molecule, available, it will slow down the speed, or the activity rather, of the rate-limiting enzyme in the metabolic pathway upstream. So for example, if you're generating a lot of ATP by going around the Krebs cycle, it would slow down the rate-limiting enzyme within the Krebs cycle. However, the opposite is also true. If you had a lot of ADP, or the low energy state of that molecule, that's suggestive that you need more ATP. And in this case, through negative feedback, that would increase the speed or increase the activity of that rate-limiting enzyme in order to generate more ATP. This is actually a really important function to have these rate-limiting enzymes. It'd be very wasteful to take the substrates that we have in our body and break them down for ATP when we're not utilizing our ATP. The primary reason is we can't store just a ton of free-floating ATP within the body. So it's important that we're only producing that ATP when we actually need it. That's why we're able to store our different substrates like carbohydrates and fats within our body to be used at a later time whenever we really need that energy. The presence of various enzymes within the blood can serve as a really important diagnostic tool in clinical practice. Anytime you have an enzyme, it's usually confined to a specific tissue or a specific cell. And it's not until that tissue or that cell is actually damaged or diseased that the enzyme then gets released into the bloodstream. It's very easy to take a blood sample and then determine what enzymes are in the blood. For example, let's say someone has had a heart attack. If someone's had a heart attack, we might see elevated amounts of the enzyme lactate dehydrogenase. And you're probably thinking, well, we're going to talk about that, or maybe you know of that enzyme already in the case of muscle. But we have an isoform that is specific to cardiac muscle instead of skeletal muscle. And when that shows itself in the blood, it's indicative that someone has had a heart attack, or what's called a myocardial infarction. Now let's say you go do your own workout, right? And you do a really hard workout. You did some resistance training, you did a bunch of intense interval drills while you're outside running and sprinting, just to pure exhaustion. If we were to assess your blood the next day, we might find elevated amounts of creatine phosphokinase, or CPK. And this is a sign of elevated muscle stress. It can also occur whenever there's some sort of crush injury or other injury to the muscle. But just you doing very strenuous exercise will cause this specific enzyme to be elevated within your blood. Now, many of you have taken an introduction to nutrition class already, but let's review the 3 substrates that are utilized for energy. We have our carbohydrates, our fats, and our proteins. So let's start with carbohydrates. When you metabolize a carbohydrate molecule, you generate about, or roughly, 4 kcals per gram of carbohydrate. It's a really helpful energy source because it can be broken down very quickly, either in the presence of oxygen aerobically or not in the presence of oxygen anaerobically. Carbohydrates are also extra important because they're the primary fuel source of both our muscles and, very importantly, of our brain. So you tend to get a bit of a fog if you're not eating a lot of carbohydrates and you're relying solely on high amounts of protein or high amounts of fat, as your body's trying to navigate how to fuel the brain under that circumstance, since it preferentially would prefer to use carbohydrates. The other great thing about carbohydrates is our ability to store them within the body. The 2 locations that we tend to store carbohydrates are in our muscles as well as in our liver. Now, you store a small amount of carbohydrate in the liver in the form of glycogen. And glycogen is just a long chain of glucose molecules, so it's a nice compressed storage form of glucose. and you're able to break that long chain apart and form individual glucose molecules again when you need to break it apart to use it for ATP. So we store a little bit in our liver, and the function of that liver glycogen is really to control for the fluctuations in our blood glucose. Whereas the glycogen that we store in our muscle is going to be broken down in order to facilitate or provide the ATP necessary to do work. So one is controlling blood glucose, right? It's fueling the brain primarily. That's the liver. Whereas the other, our muscle glycogen, is staying right in the muscle and being utilized for energy to do work. Here's the biggest drawback of glucose or glycogen. So when I'm saying that, I'm just referring to the storage form. You can only store about 2,500 kcals of glycogen within your muscles. That's not that much. And we're going to talk about another substrate that we have way more storage capacity for. Now with training, you can increase the amount of glycogen that you can store in your muscle. That's a really nifty training adaptation. However, there's still a finite amount, and you only get a small bump in the amount of glycogen that you can actually store in your muscle. So it's important that we have another substrate that we can rely on to produce ATP whenever it's broken down. Now that other substrate that we're able to rely on, that's our fat molecules. So remember, fats are broken down as triglycerides, and when a triglyceride is broken down to be used for ATP, it's broken down in the fatty acid chain, so you'll get 3 fatty acid chains and a glycerol molecule. And we'll take those free fatty acid chains and further break them down to generate ATP. Now the great part about fat is you get about double the bang for your buck in this case. So a fat molecule, you get roughly 9.5 kcals per gram of fat. So you get a lot more energy. We call that a more dense energy food if it has more fat within it, or it's just referring to a higher amount of kcals per gram of that food that you're consuming. But the key to our fat molecules is really just how much we can store in our body. On average, people store about 70,000 kcals of fat molecule within their body. That's a huge storage reserve of backup energy if we would need that in order to do work. Now the biggest caveat to that energy source is it has to be metabolized aerobically, which means we have to have oxygen available. So when you're talking about really high intensity exercise, it's not great for facilitating that. It's much better served to things like activities of daily living, right? Just your ability to go out and walk all day, or to walk around and do your work. So for example, I often tell students, if you wanted to go walk 100 miles right now, you have the calories to do so. Now, are your muscles in shape to do that? Maybe not. But you do have the reserves to be able to support these long endurance types of exercise. And that has a lot to do with our evolution as a species and what type of exercise we are more acutely attuned to be able to do. I always tell people it's much easier to do an event that's 100 miles at a moderate pace than to, for example, run with Usain Bolt running a 100-meter dash. That's a really hard feat because in general, the human body is not designed to be very fast and explosive like that. Whereas we are actually designed to do these long endurance events. And that might sound funny because you're probably sitting in your seat thinking, I am not designed for a long endurance event. You'd be surprised how your body would adapt if you started training for that. Whereas if I asked everybody to go run a 100-yard dash in less than 10 seconds, not going to happen for most of us. Doesn't even matter how much we train. We could spend a lifetime training to do that, and we still might not do it. It's just really dependent on how our physiology is tailored as a whole. Can some people do it? Sure. Can the masses do it? Definitely not. Now with that very high ATP yield, though it's great, we've already mentioned the drawback is that it's really slow to produce because we have to go through aerobic metabolism to do so. Sometimes it seems like the breakdown of free fatty acids are a lot more complex than the breakdown of carbohydrates. That's really not the case. They just have a first step that looks different by going through beta oxidation. And we'll talk about the details of this process.